All right, let's go ahead and get started. Good morning. Congrats for making it to Friday of ASHG. My name is Nina Gonzalito. I'm on the Human Genomics Segment Marketing team here at PacBio. Today, I'm going to be giving an overview of how HiFi sequencing is powering translational research. So for those of you who knew us at the last in-person ASHG, we look a lot different now as a company. First of all, we are now a nice, bright PacBio pink color, and we have grown quite a bit in the past few years. We're now a company of almost 800 global employees, and our mission statement is enabling the promise of genomics to better human health. You might know us as a sequencing company with our new instruments over there in our booth, but we do have a big product portfolio spanning everything from sample prep to software. And our core technologies are really focused on accuracy, quality, and completeness. And this is exemplified by our nanobind DNA extraction technology, SBB, sequencing by binding, short read technology, with our new Onso platform, and HiFi long read sequencing powered by Sequel IIes and our new Revio box. So in this section, I just wanted to highlight a few examples where HiFi has a lot of opportunity to give researchers like you a new look at the human genome, enabling you to look at regions you might not have ever seen before or get a more complete view of genes that are associated with various diseases, from rare disease to pharmacogenomics, transplants, neuro, et cetera. And we think this is really the tip of the iceberg. And the studies that we have done on PacBio machines were really powered by our previous iterations of our machines, from RS2 days to Sequel I and Sequel IIe systems. And this body of evidence is really setting the stage for our new machine called Revio that we launched on Tuesday. And with this machine, you now have the power to build on this evidence with scale at about 1,300 human whole genomes per year at $1,000 per HiFi genome, as well as a faster turnaround time with more compute power and a simplified workflow. So with this technology, we are really hoping to drive routine use in clinical and translational research. And some of the applications that I'm going to highlight today really address these needs in translational information. So first, I'm going to go over targeted sequencing. Targeted sequencing comes in two sort of flavors with PacBio. First, we have amplicon sequencing. We like to think if you can amplify it, then you can sequence it full length on a HiFi read. This is a more mature offering, and it's a perfect fit for any single gene assays or small panels. And because it's very high throughput, you get a very low cost. And so if you have a specific region of interest, this is great for building a fit-for-purpose assay with really low cost and supported barcoding, library prep protocols, et cetera. We have a lot of customer examples, people using this already. These are some application notes for CYP2D6 testing, thalassemia screening, and CAH screening with CYP21A2. If you go to our targeted sequencing page, linked to on this QR code, you can get access to all this material. It'll show you recent publications. One of our newest offerings is a partnership we announced this year with Twist Bioscience, using their hybrid capture probe technologies to help you design long read panels. This, again, you get your regions of interest and all the benefits of long read HiFi sequencing on a standard workflow, again, driving down cost because you can get more multiplexing on a SMRT Cell. This has supported products from both Twist and PacBio, and again, all the materials available on our website. Just a little bit more about the Twist panels. At ASHG, we announced 2 panels that we've been working on with Twist and collaborators. The first one is the long-read dark genes panel. So if you came to our CoLab on Wednesday, Fritz Sedlacek talked about this panel. This is a 22 megabase target region, almost 400 medically relevant genes that are difficult to sequence or inaccessible to short read sequencing. This is a pretty big panel, so we're running about 4-plex on our smart cells for Sequel IIe or 12-plex on the new Revio machine. And again, this is designed by Fritz and team at Baylor. We also have a long-read pharmacogenomics panel. This is a smaller panel at 2 megabases, 50 genes, as well as full mitochondrial DNA. And this includes all the major CPIC FDA genes. And it is about 24-plex or 72-plex on Revio. This was designed by Stuart Scott and team at Stanford. These will be available off the shelf through Twist next month. And if you aren't interested in those panels, it's very, very easy to design your own with the Twist team. You could stop by their booth or contact your local Twist rep. You just need a gene list or panel design, and they'll get you started. It's very quick and very easy to do. If you want to take a look and see for yourself what targeted data looks like for HiFi sequencing, we have a PacBio datasets website that has demo data on reference samples from both of these panels, as well as whole genome data from the Revio machine. Next, I just want to highlight methylation. So this feature was also unlocked earlier this year. So now with HiFi, you can get 5-methyl cytosine calling included on your reads without any additional library prep. So now you get access to nucleotides plus 5-MC. It's phased. There's no additional work like with bisulfite sequencing. You get access to GC regions and all the benefits of HiFi just with methylation. And we're starting to see some really exciting work using this data. This is a paper out of Tomi Pastinen's group at Children's Mercy looking at a myotonic dystrophy case in the DMPK region. And you see the 2 phased haplotypes here. The top one shows a CTG triplet pathogenic repeat expansion that actually induces methylation in the region next to it. And you don't see that on this other thing here. It's hypomethylated. So researchers are really starting to take advantage of this long-read technology. We have a lot of institutes and large consortia who are starting to adopt HiFi for rare disease sequencing. And you could see with PacBio, you're able to solve the unsolvable, explain more cases than you could with microarrays and short-read exomes and whole genomes, largely due to the new things you find like structural variants and methylation with HiFi. And this just highlights a few more examples of recent findings using HiFi data for rare disease. You're getting more SVs, as I mentioned, a lot more de novo variants as well. And these are all available on our website. Finally, with all this information that's being produced, we still need a way to analyze this. And so we are developing a lot of new tools for calling certain regions of the genome on HiFi. So I just wanted to highlight a few recent ones. First, we have Paraphrase. It's a SMN1, SMN2 caller for spinal muscular atrophy. It's available, the preprint is available at this QR code. We also have Pengu, which is a CYP2D6 caller that works on amplicon targeted sequencing and whole genomes. It's available at a poster just right here. And Target, which is our tandem repeat genotyping tool, Igor is going to talk about that next, so I'll leave that to him. All of these tools are available at our Pacific Biosciences GitHub repository, so I encourage you to check it out. And then right after this CoLab, we're going to be hosting a bioinformatics office hours with the computational biology team. So if you have any questions or ideas or feedback, please stop by our suite in 401. It's upstairs to the left to have coffee and snacks with the team. So next I'll pass it over to Igor to talk about repeat expansions.
Hi everyone, my name is Igor and I'm a bioinformatician at PacBio. So today I'll talk about our work on creating methods for tandem repeat analysis. If you attended my talk on Wednesday, these slides are similar, but there's definitely a lot of new content. So first of all, tandem repeats are regions of the genome that consists of tandem copies of some DNA motif. And we care about tandem repeats because they have been linked to everything from gene expression changes to cancer to diseases like amyotrophic lateral sclerosis, Fragile X, and many others. And another reason to care about tandem repeats is just, there are very many of them in the human genome. And this slide summarizes results that were obtained by Adam English at Baylor. So Adam is working with a catalog that contains about a catalog of tandem repeats that spans about 4% of the human genome. And so what Adam found is that about 75% of structural variants longer than 50 base pairs correspond to tandem repeats, right? So that's a huge number. Right, and the kinds of variation that we can see in tandem repeat regions involves everything from large repeat expansions. Some of them are, of course, pathogenic. It also involves significant sequence composition changes, some of which were recently discovered to be pathogenic as well, and also changes in epigenome, like changes to methylation. And to give you a concrete example, on the right side of this plot, we're looking at FMR1 repeat in this well-characterized sample NA07537. Right, and the way that this plot was made is we extracted pieces of HiFi reads overlapping this repeat and then stuck them together and sorted by size. And so if you look at the top half of this plot, you'll see reads that span short 87 base pair allele, while the bottom half of the plot shows you reads that span the expanded allele. The expansion is about 1 kb in length, but it is mosaic, so you can see that the exact size of the repeat changes from one molecule to another. Also, pink color corresponds to CpG methylation, so you can see that the expanded repeat is heavily methylated, just like you would expect FMR1 expansions to be. Right, and so what we wanted to do is we wanted to build a set of tools that can use data like this to analyze not just pathogenic repeats, but also ideally all repeats across the human genome. And so to this end, we developed a suite of tools. The first one is called a tandem repeat genotyping tool, or target. So this is a method that uses HiFi data to estimate sizes of the repeats, to determine the sequence of each repeat allele, and also to summarize methylation. Also, we developed a tandem repeat visualizer, or TRViz, so that's just a visualization tool for repeats. And then we are also working with many collaborators, including Genome in the Bottle Consortium, on creating a genome-wide tandem repeat catalog. And Target and TRViz are available at this GitHub link. So the way that they work is pretty straightforward, at least user-facing aspect of it. So you need to run Target, you need a set of aligned reads, so just a normal BAM file with alignments of HiFi reads. And then you also need a repeat catalog that contains definitions of tandem repeats. So on the top right, we're just looking at the definition of FMR1 repeat, same repeat that we talked about in the previous slide. So we just give the coordinates of the repeat and the structure. In this case, we know that FMR1 repeat consists of repetitions of CGGs. And then Target takes the aligned reads and then a list of repeats to analyze, and then for each repeat produces repeat lengths, repeat sequences, methylation levels, and more information. And then you can optionally use TRViz to visualize a given repeat. And I'll show you some more examples of TRViz plot on the next slide. And then I just wanted to mention that benchmarking analysis accuracy for repeats is tricky. The reason for it is that we still don't have very good genome-wide truth sets for repeat analysis. And so instead we have to do things like, to check Mendelian consistency, where we genotype repeats in family trios and then check that the genotypes are consistent. We also, we compare our repeat genotypes to the latest genome-wide assemblies and check concordance. And so I give some numbers to that end at the bottom of the slides. But if you'd like to talk about this, I have a lot more analysis that I can show you. Yep. And so anyway, let's look at some TRVS plots. So here is a TRVS plot that that shows you FMR1 repeat in a G002 sample. So the first line just shows you the repeat allele, and then lines below which show you the alignments of HiFi reads to this repeat. So you can use these plots for two purposes. You can use them to visually study the structure of the repeat, and some repeats have fascinating structure. And then you can also use them as a QC tool that to check that the repeats is genotyped correctly. Here is another example. So this is a plot of CNBP repeat region. What makes this region interesting is that it contains 3 repeats next to each other. CAGG repeat, CAJ repeat, and CA repeat. As far as I know, only the first repeat is pathogenic in the sense that its expansions cause disease, but you really need to be aware of all 3 repeats in order to accurately resolve variation in this region. Once we developed TRVs, of course, we wanted to analyze as much data as possible, and one thing that we've done is we genotyped over 1 million tandem repeats across the genome in 100 samples from the Human Pangenome Reference Consortium. And on this slide, I'm just summarizing the analysis of repeats that have some degree of polymorphism. So if you look at the bar labeled with 2, you'll see repeats that only have 2 distinct repeat alleles in 100 samples. So these are not very polymorphic repeats. While the right side of this plot corresponds to very polymorphic repeats, and the last bar shows you that a few % of repeats that we analyzed have 15 or more distinct repeat alleles in 100 samples. So these are very polymorphic repeats. And we know that most, functional, most disease associated repeats are very polymorphic even in healthy people. So the right side of this plot might correspond to repeats that are more interesting, to analyze and might be functional. And then 2 inset histograms show you an examples of repeats with 3 distinct repeat alleles and with 9 distinct repeat alleles. And in our analysis, we saw that in general, most of the repeats have like a few dominant repeat sizes, even if they have very many alleles, and then the other sizes appear at much lower frequencies. Okay, and then I also wanted to show you some results on repeat methylation. So what we've done is we've analyzed hundreds of thousands of repeats across the genome and summarized methylation levels for each repeat allele. And what we see is that repeats with repeat alleles with lower CpG density have higher methylation, while repeats with higher CpG density have lower methylation. This is not surprising at all because we know that's how human genomes are. We know that human genomes are very CpG sparse and have very high levels of background methylation. But then there are also CpG denser regions called CpG islands that many of them are in promoters and have low methylation. So this is consistent with that. But at the same time, there is a lot of variability in repeat methylation. And there are many regions that have like seemingly interesting methylation patterns. And this is one example. So this is a repeat on chromosome X in a sample from female donor AG004. And this repeat is homozygous, but if we look at methylation of this repeat, we see that half of the reads show us that the repeat is lowly methylated, while the other half of the reads support high methylation. So this looks like a nice example of X inactivation. So yes. Okay, and then I also wanted to show you some results where we applied target from data from Genomics England from 100,000 Genomes project, where we analyzed genomes of 82 participants in this project. And what we did is we targeted analysis to 56 known pathogenic repeats. And this slide summarizes our findings. So we found a methylated mosaic FMR1 expansion that spans over over 1 kb in length. We found an ataxinate expansion that was over 1.5 kb. And then we also found 7 biallelic RFC1 expansions that consisted of this relatively recently discovered pathogenic 2A and 3G motif. And some of these findings were already confirmed, and the rest are in the process of being confirmed. And then I wanted to finish my presentation with just giving you a glimpse into our current work. so well, let me start with the slide. So here we're looking at a seemingly very difficult regions of the genome, right? This is what this region looks like if you look at HiFi reads in IGV, like in the standard genome browser. And these are the alignments of HiFi reads to the reference genome, right? This is 3.5 kb region in this KCNMB2 intron. I think many people who dealt with read alignments, they would agree that this looks like a total mess. We see many insertions, deletions, mismatches, right? And when we run standard variant callers, we get, lots of spurious variant calls that are very difficult to do anything with. The bottom track also shows you existing genome repeat annotations. So this track shows you that it looks like this region contains many 80 repeats. But it's also very hard to make sense of this information because these annotations are like clustered together, they overlap, right? So it's very difficult to say anything about this. But if we kind of go back and do the sequence level analysis, and if we analyze the sequence of this repeat in the reference and also in over 100 samples, you will see that the sequence of this repeat consists of motifs that look like this. So these motifs have very heterogeneous sequence, but the same structure. They all start with CT repeat, or low complexity CT sequence, followed by this AGG course sequence, followed by AT repeat. So, and if we define tandem repeat, this tandem repeat in this way, then we can analyze it no problem. And so here is the result of target-based analysis of this region. So I'll just come back a few slides. So this is exactly the same region of the genome reanalyzed with target, right? So we see that what we see is very clean. It makes a lot of sense. We see that we in fact deal with tandem repeat. This repeat has two alleles. One of them is about 3 kb in length. The other one is a little bit longer. If you squint and look very closely at the tick marks that delineate different copies of the motif, you'll see that they're not spaced evenly. This makes sense, right? Because the motif is so heterogeneous. Some copies of the motifs are longer, some are shorter, but that doesn't matter. But as long as we think about this repeat in this way, this region in this way, I think we can analyze it, across as many samples as we'd like. And it looks like there are very many other regions of the genome that we can resolve in this way as well. And this is just an example of analysis of this region across 100 HPRC samples, and we just, this is just a histogram of lengths of this repeat, across 100 samples. So we see that some repeat alleles span about 3 kb, and then there are also some repeat alleles that span over or close to 4.5 kb in length. Yep, and that's the end of my presentation. In conclusion, I just wanted to show you links to Target and TRViz, and also a link to annotation tool for Target that we just developed. And yes, if you are interested in repeat analysis, if you'd like to collaborate, please reach out. We always love to collaborate with scientists to figure out what are the important problems that we need to solve. Thank you.